So far, on the surface of the cell, we have MHC class 1 molecules. The first one is encoded by the A0201 allotype or allele. The second one is encoded by the A3001 allotype or allele. Now let's talk about an HLA-B allele called 2705, right? So let's go back into the ER, right? Our good friend the ER, and we had this situation where we had an infection, but we're only able to present 2 out of the 6 viral peptides. If only we could present more. The more we could present, the higher our chances that we will be identified by a T cell. Well, now I'm going to tell you about HLA-B 2705. And so I'm going to tell you that the peptide binding motif of this allele is an arginine at position 2 and an arginine at position 9. What does that mean? Well, that means now that we've increased our ability to present peptides, because before we could only present 2 out of those peptides, right? The ones with phenylalanines at the anchored positions or the leucine and the valine. Well, now we have the ones that had 2 arginines, an arginine in position 2 and position 9. Well, now we can present that. So our numbers of peptides that we can present has now gone up just by adding another version of this gene. Right? So going back to here, right? If you think about the fact that you can have 6 different isoforms of MHC I, each of them presenting a different class of peptides based on anchor amino acids, anchor amino acids. In fact, you can present 6 different types of peptides on your surface. And the more peptides you can present, the more likely you are to survive the viral infection. Right? So, for HLA-B, there are in fact 872 different allotypes. So, we'll talk about the advantage of having so many different versions of the gene very soon. For HLA-C, I'm not going to tell you about any of the specific alleles, but again, HLA-C makes the alpha chain of MHC class 1, and they will present different peptides as well, 274 different allotypes. So of those 6 molecules, those 6 MHC class 1 molecules I drew in the cell, each one of them is made from a different copy of this gene, the A or the B or the C from the maternal, or A or B or C from the paternal. One from maternal, one from maternal. So what we can say about the MHC I genes, not only is there this gene family, that basically there's the A, B, and C gene, all of them encode for the alpha chain, but also these genes are highly polymorphic, right? And so this comes back to this concept of alleles, number of different alleles. The fact that there are hundreds of different alleles of the A, B gene and the B gene and over 200 of the C gene, lots of different versions, polymorphic, right? What's the advantage of having so many different versions of these genes? Well, the advantage is that you and me, and most everybody, is likely to be heterozygous for these genes. Because it turns out, in fact, it's very dangerous if you were, in fact, homozygous for these genes. And so let's talk a little bit about genetics here, about the advantage of being heterozygous versus homozygous. Right? So if you go back to freshman bio or in genetics and you think about, well, there's some gene and there are 2 different alleles to this gene. Right? There's dominant and recessive, big T and little t. Well, then what are your possible genotypes? Well, you could be homozygous dominant, or you could be homozygous recessive, you could be heterozygous, right? And this is a pretty simple, manageable concept when you're talking about one gene with 2 alleles. Well, for the MHC class 1, and let's just focus on the HLA-A gene, there are 506 known different alleles, right? So what are the odds that you have inherited 2 different ones? Well, the odds are really, really high that, in fact, you are heterozygous. It would be, very unlikely that you, in fact, have inherited 2 identical alleles, that you are homozygous. But what would happen if you were homozygous? What is the disadvantage if you were homozygous? And think about it. These genes encode for proteins that present peptides. And the more peptides you can present, the better you're going to be in surviving the infection. So if you were heterozygous, if you had two different alleles or allotypes, and you could present two different types of peptides, that's great. If you were homozygous, though, you could only present one type of peptide, or fewer peptides, and therefore less likely to show and find a T cell that will fight your infection. So back to our example of having all these different allotypes, being able to present all different peptides. So for example, for this one viral protein, before, just looking at these three isoforms, we were able to present 50% of the peptides. 3 out of 6 peptides we could present to T cells in hopes that one T cell, or maybe a number of them, will in fact identify us. Well, let's get rid of that allele, right? Let's just say that we only have the A0201, right? And let's get rid of the B1 while we're at it. Let's say we didn't even have a multiple gene family. We had one gene and not very many versions of this gene, so you're more likely to be, in fact, heterozygous. So now, the only peptide that you can present is something that has a leucine at position 2 and a valine at position 9. So instead of before, we were able to present 3 out of the 6 peptides, now we are only able to present 1 out of the 6 peptides. And that is not a good thing, right? You are less likely to survive a viral infection if you are unable to present many different peptides. So your book talks about the concept that this is actually a case where evolution has selected for multiple versions of this gene, so that's why we have a gene family, and multiple alleles, so that we are unlikely to have inherited the same alleles and be homozygous, and therefore we are more likely to present peptides. We're actually going to do an activity on Monday which will demonstrate this. So hopefully you can appreciate why, in fact, we have so many different versions of each of these genes. Right? So that brings us back here, right, where we started, which was sort of a very messy area on chromosome 6. Right? But now hopefully you understand that the HLA-A gene and the B gene and the C gene all encode for the alpha chain of the class 1 molecule. They all make the alpha chain. But what's different about them? It is very likely that they all present different peptides. They all have different peptide binding motifs. And the more different they are, the more different peptides you can present, the more likely you are to survive a viral infection. So this is how we talk about MHC diversity. When we talk about B cell diversity and T cell diversity, that fact that most every one of your B cells is different, but that was due to VDJ recombination and junctional diversity. We don't do that here, right? There's no recombination in MHC, in this MHC region, but we do inherit our diversity from our parents, right? And the fact that our parents most likely didn't have the same alleles as their parents or as their spouses, and so when we inherited our alleles were likely different from one another. So we inherited, let's say, 0201 from our mother and 3001 from our father for the HLA-A gene. And for the HLA-B gene, we inherited 2705 from one parent and the other one from the other. So the more peptides you can present, the more diversity you have in the MHC locus, the more likely you are to present higher numbers of peptides, the more likely that your T cells are to recognize your virally infected cell, the more likely you are to survive a viral infection and pass along your genes. So people who don't have MHC diversity, people who have a low level of diversity, are less likely to survive certain infections than people who have a high degree of diversity. And like I said, we will do an activity in class on Monday that will demonstrate this concept.